Hello.
Morning.
Morning. All right. So let's get started. Are there any updates or things to mention before we get started? We had our Diva Worm meeting this week, and then we had our Cognition Futures meeting. And that was our first meeting in a while. And we covered our paper on Bergson for that. So that was good. Hello, Hussain. Did you have any updates to give, or?
Yeah.
Okay.
Hi guys. Happy Saturday. Yeah. So yeah, really happy with the progress being made on the computed tomography project, and we are now getting another paper together, hopefully to get published by the end of the month. Bradley should be on board of it. It should be me, Dick, and Bradley, probably the three of us. Particularly the results of the MART algorithm. Showing how the MART algorithm can work and the simulations would go through with the context of Perlin noise. So that's why we needed your expertise on that with the Perlin noise. So yeah, I've got the plan together. I'm performing the simulations right now. Dick and I are going to be meeting twice a week in the next 3 or 4 weeks. So it's going to be a lot of work. In other news, I'm balancing the work for that startup based in California. We're not public yet, despite the fact that it's been over a year now. And that's how these things happen. That's how things happen. So we just got to be patient, keep working, keep trying your best. But yeah, that basically is what it is. So yeah.
All right.
Also working on a few other things here and there. I'm making a video game, an AR video game.
Oh, yeah.
Sorry, I'm getting aggressive. I don't know if you just want to keep me to keep it to oral stuff, but
oh no, that's fine. You can say whatever.
Yeah, thank you. I'm making an augmented reality video game as part of a contest for Niantic. So basically, Niantic, so that's the company behind Pokemon Go. They use augmented reality. They use they made like the augmented reality interfaces, which Pokemon Go was based off of VPS technology for location-based like games and stuff. And they basically updated that with Pokemon, and with adding Pokemon, be like, hey, what if we make people catch and battle the Pokemon throughout the world and stuff too? So Niantic just released their Niantic Studio, which is like their more like making one of the easier, more straightforward ways of letting people create augmented reality experiences right from their browser, from their computers themselves. Even if you're not like the most technical coder, even if you can't really afford to waste so much time coding things from the ground up all the time. And we're having a project, like a contest or competition, to make a to just use the software in the best way possible. So I'm making a game called Data Hunters, where you collect and battle creatures where their attacks and their abilities and what they say and do is based off data science and statistics concepts. I think it's a great way to like one of them would be an example of like a regression model type creature, which has attacks that are like linear or polynomial. And the different attacks and the abilities deal damage based off the concepts from statistics and data science. So like the linear attack may be very consistent in dealing damage, but it's not so powerful. Maybe it's just like a very simple, straightforward attack, like a very high accuracy attack with low variance, like a linear regression model, like maybe the variance is variant. So I think it's a really great idea to teach people about stats and math and data science from an interactive, immersive, augmented reality point of view. So yeah.
That's good.
I'm looking on.
Yeah. So that's great. Thanks for the update.
Yeah. But overall, sorry. Back to the RBRCT project. Got off the phone with the patent attorney this past week. He's very happy, very excited with things.
Yeah, that's great. I've seen you putting updates in the in one of the channels there, the one you made for the project, for the imaging project, breast cancer CT. So it's always fun to watch those updates.
Yeah, exciting stuff. Really a lot of exciting stuff. More importantly, just like for me on a personal level, I'm really proud and happy I've been able to get this together, to be mature, to be confident, to be regulated about this, and to really more or less own this as what I'll be doing going forward. So yeah.
Okay. That's great. Yeah. So this is the LearnMem conference. This is from 2023. This was held in Huntington Beach, California, and this is I think this was associated with the Bergson paper in Cognition Futures, because we were talking about memory, learning and memory, mainly where memory is stored in the brain. And the typical model is where you have, the file drawer model, where the hippocampus is involved in looking things up in the file drawers of the parietal cortex. And this paper that we were reading argued that actually, if you take a ecological psychology perspective, it's actually not. You can actually link memory to action and to sensory processing. And so it was an interesting take on it. But anyways, so this was a conference, I think the most recent LearnMem conference. And I was just looking through, I was wondering about the state of the field in terms of like what learning and memory, what's going on in that field. And so this is something that's hosted by the Center for the Neurobiology of Learning and Memory. And so, that's where it was held. And then the program, let's see, we'll look at the featured speakers. So, we have a lot of speakers here on different topics. Anesthesia, associative learning, frontocortical circuits and reward learning, sex differences in learning, targeting metabolism to enhance cognitive resilience during aging, how memory guides value-based decisions, how moments turn to engrams in the brain, prediction error and memory updating, and so forth. So they have some pretty good, also affective learning as well. So they had some pretty good topics there. So yeah, this is sort of the state of the field. So this was, I think Morgan put this in the Slack. So thank you, Morgan, for pointing that out.
Well, yeah. The paper actually had video links to the 2018 meeting.
Which paper was that?
The Bergson.
Oh, okay.
Or the one that we covered. Yeah.
Okay, so they actually cited the meeting. What was the context of that? I can't remember.
One of the speakers' videos was actually a reference in the paper.
So this is actually Robbins is the author, and then it was the Bergson memory paper. Boom. This is it here, yeah. So this is this is the abstract. So this is is experience stored in the brain, a current model of memory in the temporal metaphysic of Bergson. So this is Stephen Robbins. And then talking about Bergson, this is Axiomathus, which is, I guess, a philosophy journal. So this is, kind of talking about actually linking memory to consciousness and experience. So it's interesting how, they're kind of taking a different view from this, the things that you might find at this conference, which are very much, clinical and, in the sort of cognitive science milieu of learning and memory, and the reinforcement learning type approach, and kind of expanding it out to this more philosophical and consciousness-based approach. So that's interesting. Yeah, and it's okay. We don't need to get into the paper. I think that's that's good. So I talked to Stefan Dvoretsky this week in an email, and he sent me a link to a group at the University of Illinois. I wasn't familiar with, I was kind of familiar with their work, but not their initiative that they've started out this year. So this is a New Scientist article on this work. So Stefan Dvoretsky he used to do, when he was in the lab, he did a lot of work with Bradenberg vehicles and, the stuff that we were doing, developmental Bradenberg vehicles back in 2019, 2020. And he's since gotten, an advanced degree in bioinformatics, and he works in Germany in bioinformatics and biomedical, science. So, he's done he's put out several papers since then. And he was interested in this project here, which I'm going to talk about now. So, this is an article 80,000 mouse brain cells used to build a living computer. So, this is a microelectrode array that is where they grow the neurons in a culture, and they allow the connections to grow, and then they use it for computation. They stimulate the cells, they record the output of the cells, and they can basically do all sorts of computations with it. So this is where you have an array of 80,000 mouse brain cells, and this is where they use it to build a simple computer that can recognize patterns of light and electricity. It could be eventually used in robotics. So I don't have the subscription to this, but I'll just read this first paragraph. So a computer built using tens of thousands of living brain cells can recognize simple patterns of light and electricity. It could eventually be incorporated into a robot that also uses living muscle tissues. So you could map the signals to muscle tissues in a robot and provide feedback from the environment, and use it as sort of the brain part of that sensory motor loop. Artificially intelligent algorithms inspired by the brain, called neural networks, have been used for everything from chatbots to searching for new laws of physics. Normally, these algorithms run on conventional computers, but Andrew Dow at the University of Illinois and his colleagues wondered whether they could instead use actual living brain cells. So that's kind of the project. And so this leads to this group at the University of Illinois. This is funded by the NSF. This is MindInVitro. And so they actually have, it's a group of researchers. I don't know how many universities are involved, but I think mostly at the University of Illinois. And they have, this is sort of their experimental setup where they grow the neurons in an electrode array, and then they record from the neurons and they stimulate the neurons. And they achieve this closed loop feedback between the neurons and the environment, which is basically whatever they're stimulating it with. So you could use, say, like a video game, or you could use, pulses of light, or you could do a lot of different types, set up a lot of different types of closed loop systems to, do these computations with. So their sort of statement here is, can computing systems be built out of living neurons? Can they achieve basic hallmarks of cognition, such as learning, attention, curiosity, and creativity, so pervasive in biology yet elusive in modern computing? And then they talk about how, can we build these kind of neuronal computers that can be programmed instead of just stimulated and recorded from? So this is another thing we'd like to do. And we could have a system that grows, heals, learns, and explores. And so that's, that's kind of the goal here, is to build that kind of system. And then by shifting from digital rigid architectures to plastic cellular substrates, we trade off speed, accuracy, and exactness for statistical robustness, extreme parallelism, healing, growth, and superior energy efficiency. So they're kind of identifying this trade-off between the things that are associated with, I don't know if they're associated with neural networks, but when you compare them with, like, brains or populations of neurons, we have speed, accuracy, and exactness. You get exact solutions that are optimal. You get accuracy and you get speed. And then
it's always difficult. I think it's always difficult in many ways. What exactly you're referring to, right? I'm not even trying to get pedantic about semantics here. It's really like especially with terms and terminology from that. Which direction are you going in? Say, I'd like to see more on that. Basically, what I'm saying is, are you optimizing from a biological standpoint to be more artificial, or in the other direction, right? So I appreciate that, and I think the more unified term for those more general concepts that can be translated more easily and effectively. There's like it's tough. It's tough, right?
Yeah.
Like so yeah. Like going from the balancing act or the like a seesaw, one takes away from the other to something where they both
Right.
it's more altruistic, more dynamic, more yeah.
Yeah.
So cool stuff.
Yeah. So yeah, that's basically, and then so they say rather than Boolean logic, these post von Neumann systems will harness the compositional dynamics of billions of neural elements to generate out of the box emergent forms of computation. So that's the other thing, we're dealing with a von Neumann architecture in the artificial systems and a yet to be determined computational paradigm for the biological networks. And so that's basically what they're doing. So they have that. That's basically they're about. And then the people. So there are a bunch of people involved in this. A lot of people from the University of Illinois. John Beggs from Indiana, and Nicole Martinez from Stanford. Another person from Stanford.
Ben's was great. Ben's was great. I took a couple physics classes under him. So yeah.
Okay, yeah, that's good.
Yeah. Is he still in Indiana?
John Beggs? Yeah, I think so.
Okay, so he's still in. Cool. Yeah.
Okay.
Cool.
So yeah, and then one of the people here, the PI is Mattia Gazzola, and so Mattia Gazzola does some really interesting work in sort of mechanical engineering and building soft interfaces, biological interfaces. He has a really nice website here. Basically he's doing all this stuff. He's doing the MindInVitro stuff. He's doing the stuff with viscous streaming, which is where they, like put things in fluids and do some engineering around that. The flow numerics, this is numerical biophysics. And I'm trying, this one is the soft creatures, which is actually a set of publications they've done on this soft robot, which basically is analogous to an octopus. So they have this octopus robot that has is made of this soft robotics paradigm where they use soft materials. And they, I guess, eventually want to put this sort of artificial mind in it, or artificial brain that has, biological properties. So that's really interesting stuff. Thank you, Stefan, for pointing that out. I should have known about that more, but I'm not familiar with this group so much. So yeah, that's good.
Do you mind dropping a link in the chat for that?
Oh, for which link? Oh, the center?
Yeah, please.
All right. Yeah. So I'll drop a link for the center. And then drop a link for the Gazzola Lab. Actually, I'll just put in maybe the one for soft creatures, and then you can look at that website. So those are the two references.
Yeah, great.
So Morgan, have you fulfilled your tooth fairy duties?
Yes, but now I'm just well, now I realize that my wife had actually done that. And,
Okay.
We had a miscommunication last night. So but yes, all settled. Give me. I need a few more minutes. Yeah.
Okay. Yeah. So let's see. Yeah, I guess Jesse will be joining us here. So going back to the Slack, I know that we're under the tyranny of Slack and their parent company in terms of having access to archives and things like that, and I haven't been able to build a proper or get a proper sort of nonprofit status. Yeah, I know, but it's hard to migrate everything. But anyways,
Slack was supposed to replace email because people would spend 2 or 3 hours a day checking their emails instead of getting work done.
Oh yeah.
So they're why don't you use Slack instead? You know what people do? They spend 4 or 5 hours in Slack every day now. So they didn't solve the problem of email. They just made it even worse, because I would procrastinate so much more in Slack compared to my inbox. So
Well, yeah.
Yeah, it tries to be professional, but it lacks the functionality of Discord, and it's yeah.
I think Discord emerged because of gaming, so it was like kind of, you wouldn't expect a gaming tool to be more efficient than the business tool.
Well, I mean, if they wanted it for gamers who play video games in real time, you cannot afford, for example, any sort of latency, any sort of suboptimal performance. So Discord was already engineered from the get-go with that in mind. Slack was more so like, hey, we want something to be professional, to present to workplaces, to things like that, and it ended up just lagging behind in also functionality. So the game developers, like, they made something better.
Yeah. So so I'll go to and I've been promising to do this for a while, going to the Brain Organoids channel. And the Brain Organoids channel, we've got some pretty good stuff in here. We haven't talked about it in a while. So
sorry. Yeah, but my apologies though. I will say that Celsius's ORAC channel has perhaps taken over in terms of the organoid references.
Oh, okay. Well,
I mean, there's good stuff there for sure. I would just want to add Sergio Pasca at the Stanford Stem Cell Center has a new paper. I'll find a link just one second here, but
all right.
Has a new paper with a four organoid assembloid.
Okay.
And which could be a new record. Anyway, but I'll add a few links from ORAC just to make sure that we've got it kind of fully synced.
Okay, yeah, you could put it in the channel, and then by the time you get it in, we'll be down there. So, okay, so this is LinkedIn, James Glazier on LinkedIn. James Glazier, of course, is from Indiana, and he does a lot of the multi-scale modeling and the CompuCell 3D stuff. And we've talked about this a lot in DevaWorm. So this is, kind of DevaWorm related, but this is the talk series on multi-scale modeling in viral pandemics. So they do this Glimprint series. And so I guess these are announced on this LinkedIn page. And, this is an interesting set of talks. We've talked about a couple of specific talks in DevaWorm. So, if I'd like to actually explore it more. Just, haven't had the time to kind of get my head around like some of the talks. But yeah, so that's that's a good resource for looking at. Actually, in DevaWorm we talk a lot about multi-scale modeling and biophysical modeling, and so Susan is interested in that, and of course Morgan as well. And so that's something that if people are interested in collaborating on with respect to building models, that would be something we'd be interested in. We've also tried on a number of occasions to try to build models in CompuCell 3D, and I know Susan went to the CompuCell 3D thing this summer, and so we've never been able to get that off the ground. I remember visiting the Biocomplexity Institute at Indiana almost 9 years ago now to give a talk on DevaWorm, and we haven't really ever been able to do anything with CompuCell 3D. So
yeah, it's been a struggle. See, because I met Glazier back in 2017. I took a class under him, like 2016, 2017. Speaking from my apologies for interrupting you. Speaking from CompuCell 3D's point of view, there has been a lot of progress that's been really slow, if that makes sense.
Yeah,
and things were tough. I know, like he himself, I don't remember specifically what it was, whether it was like funding or just like the overall directions of the project and where things are going. And a lot of researchers may be getting a bit frustrated or with the direction of things in some ways. So like it was what it was. I remember I took a class under him, which was basically about using CompuCell 3D.
Yeah.
Like I think it was like some like computational class, like something like that. Yeah. Probably something similar. Like but yeah, he it was what it was. It was him and Robert DeVoider from the physics departments, and there were just many differing takes on where things were going. That's what I particularly remember. So yeah, the Biocomplexity Institute, and where things went with that, or how the professors and the faculty were there with that. That was an interesting one too. That was a great one too. I also remember like there was a lot of movements from physics to the new engineering school. There was that going on too.
Yeah.
So IU Bloomington started their engineering school back in 2016. Yeah.
Okay.
Fall of 2016. And to differentiate it from Purdue, which, it's like, oh, you're in Indiana, you want to study engineering, you should go to Purdue, or like Rose-Hulman or IUPUI, somewhere similar. IU did not have an engineering school. IU Bloomington was more known for like liberal arts or like basic sciences, like life sciences, like physics and stuff.
Right.
But the engineering school itself, there was a lot of push for that. There was definitely a lot of push for that. To make it different from what Purdue offered, but still make it like really appealing. Like there was like neural engineering, there was like bioengineering, which would be different from like BME, biomedical engineering, which is what's offered at Purdue. So there was definitely a lot of discussion on where things like CompuCell 3D and the Biocomplexity Institute would fall, and I think there was a lot of confusion with some things there too. So meanwhile, I'm just like, studying physics and just trying to graduate and see what I want to do next. So yeah. But yeah, that's basically what I remember from how things were about seven or 8 years ago. So yeah.
Well, I mean, we've had like people interested in it, and there's sort of like a learning curve to any modeling platform, and then you have to find a good problem to apply things to. So that's always the challenge in doing like a lot of good simulation work, is having the right kind of representation, the right problem, and then apply, getting it to work.
I mean, even in my experience, from my background, coming from someone who studied bioinformatics, there was just a lot of hype for things like the Human Genome Project, and a lot of things really did not meet that. And I feel like maybe in terms of the computational scientific research world, people began becoming very risk averse to just posing deep questions like, hey, can we just mathematically simulate how an octopus moves and use that to apply to everything? And they're like, you want money to perform simulations and work with data, that's great, but you have to be realistic. It's more so about like how realistic things are and how trustworthy people are with like giving funding and devoting time and energy to stuff like that. So yeah.
So that's that. And then we had this paper here, which is towards learning a foundational representation of cells and genes. This is Mohammed Latfolani, and this is just, I guess, an opinion piece on how we can build. I guess they're going towards foundational models of some type, where you're trying to learn this foundational representation of cells and genes. So one of the problems in biology and systems biology, but also in like applying machine learning to biology, is this idea of what is the representation we should be using. So there are a lot of ways you can represent, say, a cell or the relationship between genes and cells. And so they're kind of giving their take on this, which of course involves some sort of transformer, putting in like having a single cell and having a bunch of genes, and then running a transformer, finding a gene embedding, which is basically where you've got similar genes and dissimilar genes, and then cell embeddings where you have different cell types. And so then you have these embeddings and you can map, I guess, one to the other. And, you're training it on data, I guess, physiological data, and out pops the, and this is all based on scRNA-seq. So you're dealing with like single cell transcriptomics, So you have very precise data with respect to single cell types. So you can build, representations from single cell types of the genes involved in whatever. You have similar genes. And then you have these cell embeddings, which are all of the single cells that maybe constitute a brain or a liver or a lung. And then you can build from there. So it's really kind of an interesting take, although I think it's that's a long-term vision, because as we collect single cell data. We have a lot of single cell data out there now. The question is can you make sense of it? And that's been, usually when you go to a paper, they usually give you a UMAP, which is just basically like this pretty picture. And then sometimes, we have a lot of information about sequences and their expression, but then, trying to figure out the relationship, like these genes that are similar, should that be represented as a network, or should that be represented as like some sort of other model that, like a model of relative contributions, or what exactly is the model that we want to use to represent this this relationship? And that's still unclear. It doesn't matter, your kind of latest approach. You just have to have something that can maybe predict the phenotype. And that's so it's an interesting take, but it's prefaced with the caveat that people have been trying to do this for a while. Like Hussain said, the Human Genome Project, the idea was to get a draft genome and then to use it for biomedical advances. And so
Yeah, I mean,
now,
my takes, I really don't mean to sound pessimistic or cynical, but like, it just really fell short time and time again. Like Collins was, like Dr. Francis Collins would hype it up, and people would pour loads and loads of money and stuff into it, and it turns out biology is very complicated,
Yeah.
and I think people really, really, really misunderestimated like what things were capable of back then in the I mean the at least like the human brain project from a comp neuro point of view,
Yeah.
which did fall into many similar struggles over its course. Like, yeah, I mean it is what it is. It's best to just like just like take steps back from things every now and then, and see if you can really refine your approaches from the ground up before getting back into something. So that's what I'll say. Yeah.
All right. It's like Morgan, maybe it's something to say, or
biology is complicated.
Yeah.
Completely agree.
Yeah.
I mean, biology being complicated is part of that, but more so, like reductionism itself, that everything is applied physics and applied math, or I don't know, like scaling things up from a cognition and from a cognitive point of view. The ability of humans, for me and you, to understand not just the equation that I'm solving right in front of you, but what does it mean in practice? What does it mean in practical applications and scientific research and theory, and how well things can actually move forward? That ability for that sort of flexible thinking, I feel like is not taught or instructed nearly as well as it should be, even for people with PhDs and MDs and all the Ds and stuff. Like you can be. What I'm saying is as good as you are as doing scientific research, publishing papers, showing great amazing findings, how well those are put into practice, even with something as big as like the Human Genome Project, is a completely different story. So yeah. So
yeah. But it was a very important project, right? I mean, it had to be done, and once it was started, yeah, like the reason I'm just adding this is that not recent, but this year, the director of the schizophrenia research group outside of DC. I'm blanking on the particular group, but but he had this big editorial about, the Human Genome Project was a failure. And
Oh yeah.
Yeah. I mean, everyone has very strong opinions on it. Yeah.
But and it's just I think he was conflating, a lack of progress in psychiatry with somehow, the Human Genome Project being at fault.
Right.
And I took some umbrage.
Failure is a strong word. Failure is a strong word, but on some level, there was a lot from an epistemic point of view that was more or less taken for granted on how things would scale up, how we could reduce things properly between different contexts, how sort of computational approaches could generalize. Like, we've got so much computing power, mathematical power in facility. Why not just shoot all the data in as much as we can? And
I mean, I don't think it was computational. I mean, again, I think it was really interesting how Ventnor went his own way and what he did to make the project happen faster.
Yeah.
Right. and some of those are still with us today.
Yeah.
Right.
Yeah.
Like Galaxy Project and other things. Certainly, he was one of the drivers behind a lot of distro development for scientific Linux. but if we hadn't done the project and kind of had all that data out there, then maybe people would still be talking the way that they were talking before the project was done, right?
Yeah.
And now it's like I think it's a good thing that sort of it opened the door to this new respect for these additional layers of complexity that are actually so much greater than just this sequence of letters.
Yeah. Right. And not even also, excuse me for getting political, but even what people what Obama would be saying too, some of the claims he'd be making about the promises of precision medicine, of the Human Genome Project, and how much hype he was able to generate through things like that. But I always think of something from, once again, my apologies for just bringing politics into this, but it was meant to be something he generated appeal and everything around. So it's like fair to be like, well, where is it? Where's the change? Has it happened? And
Right. But that's again, this particular editorial brought that up. I think brought up Clinton. Because of course, Human Genome Project was more his era. Or
it was more like Clinton and Bush era. It really was. And the Human Brain Project was more, the Brain Project was more Obama's era. So
and he was just cribbing from their grant application, So sorry. So
no problem. I mean, it is political. Like as much as Dr. Francis Collins wants to argue he's a scientist, he's so pure and so noble his intentions all the time. The dude has a band called the Affordable Rock and Roll Act. Like you can't talk around that. And I'm sorry because I met Collins. I met Collins a long time ago.
I love all these band names.
What the hell?
What's Earl Miller's?
You tell me like these projects are not political. You tell me they're not political, at least on some ways. But I
have,
sorry.
Because of politics and it seems like all my favorite podcasters are all former speechwriters for like Obama or Clinton.
Yeah.
I've gotten to hear more about how the sausage is made.
Yeah.
Excuse me. I mean, but I say this for a reason. With Obama, from his first term to his second term, people on some level losing trust in him, becoming a bit disillusioned, and saying he promised so much change. Like, are things happening? A lot of people are still struggling with unemployment. Back in like 2008 to 2016. There was the recession, and people were like, did Obama really follow through on things? And one thing I'll say, and this is where it comes into the context of the Human Genome Project, is a lot of times it's like you can't always wait for someone to change things for you. You have to show the change yourself. And whether or not that applies to the Human Genome Project, like with Obama saying, yes, we can change, yes, we can fix things. He wanted that support from not just for people to vote for him and then sit on their ass and do nothing, but like, in terms of research and scientific change with the Human Genome Project, we have to be like, we have to be mindful, like we have to be like we are the ones implementing the change. We are the ones making those discoveries. It's in our hands. So yeah, that's what I'll say. Yeah.
I think.
Well, yeah. Sorry, go ahead, Bradley.
Well, one of the things, we confuse kind of scientific infrastructure with and this is more generally like the way science is almost viewed nowadays is that we confuse building infrastructure with like making progress on scientific problems. And you see this all the time, like with like applying computational methods to things, and it's magically going to help you discover things that you really need to have good theories, or you really need to have good experimental synthesis, or even just good experimental methods to get at. So if you build a piece of infrastructure that brings information to people that they can use or they can draw from to solve their problems, that's great. But that doesn't solve the problem. You still need to have that other part. And we fund a lot of like tool building, but then the tools get kind of, we get a lot of tools and we don't get any insight because people aren't funding that, or we just think that like just building the tool will magically lead to the insight. So
I was also just thinking of this this paper that recently I can't exactly remember the conclusion, but it was analyzing, the text analysis of journal articles in the past 20 years or something like that, and the decreasing use of something like the abstracts and conclusions using stronger terms for certainty and things like that. Like just thinking a bit about the rhetoric of science, the rhetoric of science, and again, kind of like what are the incentives for that rhetoric? The dangers of falling for our own rhetoric. Perhaps.
All right, let's go through the channel more. So this is, I guess, another LinkedIn link. This is a public version of the talk slides that this person gave, Jian Ma, at UCLA. This is large language models and computational biology, a primer. So this is just kind of talking about. Yeah, go ahead.
Yeah, I was just gonna say, if I remember right, this was there was two things that were happening. One was an IPAM meeting that was interesting, and one was like computational genetics, like seminar series, but like they'd obviously had a workshop because they dropped all these videos at the same time.
Okay, yeah.
But really, like definitely, sometimes when I'm dropping these links, it's they're kind of representative of a kind of a larger meeting. Yeah. So I don't know why that's just an image.
Because I didn't go to the LinkedIn site. That's okay.
Okay.
I just wanted to.
Yeah, sorry.
Yeah.
like I've got issues with X.
Yeah,
Twitter.
I know. But yeah, I didn't want to open up LinkedIn because yeah. Anyways, yeah. So that's what's going on, sort of with these kind of foundation models in computational biology, and certainly that is something that. We have the Human Genome Project. We just have kind of like the sequences, some of the comparative work, the tools for the comparative work. But then getting to the representations getting to the actual comparative work, the science, figuring out like what, I don't know, the funding agencies like to say the rules of biology. Now, whatever those are, those don't come through magic either. We need to kind of do the work to find out what those are. So that's all kind of built on top of our tools, not tools as a sort of a replacement for that. So
I'm just saying that in that link, he does actually have links to both his slides. It was the seminar, the computational genomics seminar, and he's got links to his slides both from this year and last year.
Oh, good.
So I'm just gonna check, and I'll try and post the actual links.
Okay, that's fine. Yeah. So these are definitely like his work in progress, which is good because this is a pretty fast-moving field. Like
for sure.
Yeah.
For sure.
So there's this bioRxiv paper. This is.
I don't know why I didn't grab the. Oh yeah.
Yeah. So this is Spatial Dynamics of Mammalian Brain Development and Neuroinflammation by Multimodal tri-omics Mapping. What is tri-omics mapping?
So it's multi-omics, but obviously 3 in this case.
Okay, yeah. So the abstract reads, the ability to spatially map multiple layers of the omics information, which is just like where you do one of these next-gen sequencing, use one of these tools and you get like whole genome sequencing and expression data using, and so there are all these different methods that people use. I guess there's RNA protein seq, there's genomic seq, you can just get all these different types of information, and then combining them is the multi-omics aspect. And then
definitely going for this immunofluorescence imaging.
Yeah. So you can build multiple layers of a biological model. So like genes, RNA, protein. You have different time points where you can look at brain development, differentiation, arealization, and alterations in disease. They develop and apply spatial triomic sequencing. So this is bit-ARP-seq, spatial ATAC RNA protein seq, and DBIT CTRP-seq, spatial cut and tag RNA protein seq. And then together with multiplexed immunofluorescence imaging, which is Codex, they can map spatial dynamic remodeling and brain development to neuroinflammation. So they're getting at this problem using all these different methods. And then they have a spatiotemporal triomic atlas of the mouse brain. This is obtained at different stages from postnatal day P0 to P21. And then this was used to compare the regions of interest in human developing brains. So you build this atlas in the mouse, use these techniques to build up information about this, and then you compare it with human brains for similar stages. And then you can find that specifically in the cortical area, we discovered temporal persistence and spatial spreading of chromatin accessibility for the layer-defining transcription factors. So they were able to look at like chromatin and transcription. In corpus callosum, we observed dynamic chromatin priming of myelin genes across the subregions. Together, it suggests a role for layer-specific projection neurons to coordinate axonogenesis and myelination. We further mapped a brain of lysolectin neuroinflammation mouse model, which is LPC, and observed common molecular programs in development in neuroinflammation. Microglia, exhibiting both conserved and distinct programs for inflammation and resolution, are transiently activated not only at the core of the LPC lesion, but also at distal locations, presumably through neuronal circuitry. This work unveiled common and differential mechanisms in brain development and neuroinflammation, resulting in a valuable data resource for these things. And so this is, again, kind of like building a tool, looking at the data, finding some insights, and that's kind of the way we do things. Now, there's probably a role for synthesis of a lot of these studies where you can actually get, because you're making some limited insights here. You're collecting a huge amount of data and building this tool, and so and then making the cross species comparison. So, this is really kind of the way we do things, and like you see, like where kind of the promise is. We're not quite there yet.
If I could just add a little bit about the previous the previous link, the
this one here.
Yeah, the large language model stuff.
Yeah.
Just to say that this Computational Genomics Summer Institute, and it has a website, which I will drop in as a reply here, and it has it has saved all the previous talks, videos from all the previous years, every single year going back to 2016.
Okay, yeah. That's
and just something that's like Stanford recently did a foundation model. Michigan did a foundation model summer school, and just because I know we brought this up, someone was talking about foundation models, and they were like, "How many people have read the Stanford white paper on foundation models?" And and not enough people raised their hand. Well, not enough people raised their hand, and he's like, okay, well, it's a very long document. How many people have at least read the introduction to? And that's only 10 pages, and not enough. And he was just like, okay, if you get anything from my talk, read the first 10 pages of that. And just to be, he thought that they were very clear about what the definition of a foundation model was.
Oh.
I think Jesse had posted something about frontier models, or anyway, that we've maybe anyway. It would be good, and yeah.
Okay.
Just wanted to say. Plus, this looks like a really great reference in terms of the I'd love to see this next year, and gather together a better, start to kind of mine our calls for participation links, to mine these kind of like annual events that we should have on that calendar to at least be an attendee or something.
Yeah, I think that would be good. I know that like there are so many events, and it's like there's so much sort of good information in them, but it's hard to get them out and say, "Oh, this is what all these groups are doing." Yeah,
for sure. And but that's the thing, right? That some groups focus on kind of trying to distill down like that progress that year, from get some representative researchers and get them to do some of their own distillation. All right.
Yeah, so that's good. And then this is the Computational Genomics Summer School here in Bioinformatics. This is the UCLA event. So this is the site for that, and there are different, different years that they have for it. So that's good. Thank you, Morgan, for that link. And so then there's this link, researchers to investigate genetic roots of autism, look for new treatments. That's, of course, related to computational psychiatry as well.
Yeah. As well as being some news from the Brain Engineers group.
Oh, okay, that's Brain Engineers.
Yeah. But that's cool.
Yeah. There's this link from X, which is, or Twitter or whatever. Exciting new inroads into human biology and therapy development. Human intestinal organoids with an autologous tissue-resident immune compartment. And so this is the image. I'm not going to link through to the post, but this is human organoids with an autologous tissue-resident immune compartment. So they have, of course, this autologous tissue, and so they're creating this human organoid, and they're able to do the stain to show this immune. I guess it's a sort of the origins of an immune system, because you have immune cells and you have the epithelial cells surrounding it, and so we don't have an immune system yet, but we have an immune response of some type going on in the organoid. Yeah, and so that's just, again, this sort of work where we don't know exactly where it's going. It looks promising, but we don't know. Obviously it's not an immune system, but it's something.
And of course the other interesting thing about these kind of gut organoids is that you can also have neurons.
So that's then there's this paper, Profiling Cell Identity and Tissue Architecture with Single Cell and Spatial Transcriptomics. This is, again, another spatial and single cell transcriptomics paper where they're,
and we were kind of pushing at Celsius spatial transcriptomics, and again, and partly that's just where things are in terms of kind of technique complexity at this point, or advancements. But yeah, really interesting, and like probably more papers. Again, like this absolute flood of papers, and trying to find good papers to kind of review all the information that's coming in right now.
Yeah, it's definitely flood of papers where they're trying to do stuff like this. So this is
this is a review article.
So this is where they're using single cell transcriptomics. This is this next gen sequencing, and they're trying to do this in the context of cellular diversity and gene expression dynamics in healthy and diseased tissues. So comparing healthy and diseased tissues, looking at the different cell types, looking at gene expression dynamics. And we haven't talked about it in a while, but in Diva Worm, we were talking about this for a while, and we haven't talked about it, but that there's this large discrepancy in what constitutes a cell type. And so, people have estimated, say, for example, all the different cell types in the human body, all the different cell types in the brain, and you can, of course, identify cells, but like if they have a clear type, and identify those, but it's always an exercise in classification. So sometimes cells are incompletely differentiated. Sometimes they transition between differentiated states. Sometimes there's states that are cryptic, so you can't necessarily know if it's a different state or not, from some other cell that's very similar. Sometimes, it depends on how you define state. So it could be largely functional. It could be based on the sort of the morphology of the cell. It could be based on sort of the class of the cell. In other words, if it is like comes from a neuronal precursor versus a muscle precursor versus like some sort of epithelial precursor. Those are all different sort of, cell types, archetypes, and then you get different cell types underneath that. Furthermore, you can have cells that undergo, apoptosis, which is programmed death, or they might be in some sort of like precancerous state or cancerous state, and other types of intermediate states of dysregulation, which is basically why cells kind of pop out of their state after they're differentiated. So there are a lot of different things going on, and you know, it's always worth thinking about these technologies with respect to that problem, which is estimating how many cell types there are in a tissue or in the body, and then what makes a cell type. So here they're, kind of thinking about, you know, like looking at cellular diversity, which means that they're trying to do some sort of census, and then they're looking at the gene expression dynamics and how those change between healthy and diseased tissues. So in diseased tissues, we expect there to be more dysregulation of gene expression. Healthy cells or healthy tissues, we expect gene expression to be more typical of a cell type. So that's the idea. And then, this is of course a review. So we, with this kind of data, we can provide new insights in developmental hierarchies, cellular plasticity and diverse tissue microenvironments, and spur a burst of innovation in computational methods for single cell analysis. So again, we're back to sort of tool making, tool building. But there's a lot of stuff going on here. There's been a lot of progress in data integration, identification of subtle cell states. So, I talked about trajectory modeling. So how does a cell go from a stem cell to some differentiated state or to some other differentiated state? Deconvolution and spatial analysis. So there are a lot of things here that, involve sort of the application of methods, but also the basic biology. They're interested in foundation models and how you fit single cell and spatial transcriptomics data into those foundation models. So this is really kind of a nice review. I don't have access right now. I don't know why. Well, anyways, so yeah, this is a nice paper, a nice review article. So then we have some more here. We have this, I guess this is a thesis towards a general framework of multicellular systems biology with application of cellular motility and phenotype control. And then this is the Simons Foundation newsletter. So this is kind of, I guess this is on some sort of modeling that's going on.
Yeah. I will say, and I mean, they had their general meeting, or I forget what they call their meeting, but I think in bioevolution I put a link that my friend sent me on the geometry of multicellularity.
Okay.
That was a really interesting talk from this theoretical biologist at Cambridge that was really interesting. What's this species? He was pointing out that Leeuwenhoek had described this species in 1700, and multicellular.
Volvox. Yeah, I see.
But yeah, exactly. And just some interesting stuff there. Yeah. Okay. Oh, you got it. Okay.
Yeah, it's right there. Yeah.
Yeah.
So this looks interesting. Yeah.
Really, really nice talk. Really nice talk. And he's talking about things in kind of the way the Glazier and modeling does permit. Yeah. But obviously very simple organisms.
Yeah. So that's let's see. I'll go back to where we were here. Okay.
Yeah, there you go.
So this is Jesse. He wanted actually to talk about putting together a general project description for brain organoids for some sort of space or project or channel. So I don't think we ever discussed that properly, so I don't know. I know we've been preoccupied with the ACT-INF or symposium, so maybe after that passes, we can get into that.
Yeah, I'd really like to go down to Santa Cruz and meet with brain engineers and see again, like I've made lots of contacts, but really try and put it together in terms of, how much we can build on what they've done, and kind of which directions would still be, kind of interesting to move forward. Yeah, which directions that kind of like needs more work.
Yeah, I think that would be good, and defining like what those things are, and kind of figuring out how to do them.
Absolutely.
Okay.
Okay.
Yeah, Jesse might go to California for business.
Yeah.
So this is a paper on non-invasive quality control on organoid cultures using mesofluidic CSTR bioreactors and high content imaging.
Yeah. So this is again trying to get at a little bit around the equipment, right? Yeah. And just like again, if we're going to do it, we want to be able to do it at a scale that's an impressive scale, and being able to control the organoids and keep them fed is key. And it also speaks to, if you can start to use alternatives to centrifuges, then you can potentially this would help in terms of having, let's say, I don't want to say different, like different spatial structures could be encouraged.
Yeah.
Does that leave me enough room?
Yeah, I think so.
But you know what I'm saying, like as opposed to just getting kind of spheroids,
right? Right.
And this will relate to a later kind of link on Matrigel.
Okay, yeah. So getting the shape is important, because the shape, of course, is going to determine how the cells behave. Basically, if you're within a ball versus,
right. I mean, again, when you start to think about embryonic development, and again, like how genetics both controls development and yet doesn't have a spatial plane.
Yeah.
Like, or at least is the spatial plan is still very is more kind of revealed. That so much of it has to do with just spatial context, and like at that moment, what are your cell neighbors, as well as what are the physics forces, like compression and shear, like mechanical forces on those cells, right? That's the really other interesting part of embryogenesis, if you will.
Yeah. So we in DivaWorm we've talked about sort of these models of tri-junctions and 4 junctions and 5 junctions, and this is where the cells are in the sheet, and the sheet is the interactions between the cells are such that they form these complex junctions. So the sheet, where like maybe 5 cells interact or 3 cells interact, and there's been a lot of emerging work on tri-junctions in particular. We have 3 cells that kind of meet at a point, and then they're sharing forces basically at that point. I mean, directly. Yeah. They usually share them indirectly. And so,
no. Exactly. And there's some, this kind of mechanobiology as a field, trying to get at, trying to make non-invasive measurements of physics forces, and to learn more about their impact. I mean, I've previously seen some of this in terms of it's discussed a lot in cortical sheet development, like why we have the convoluted brain, and the kind of mechanistic theories behind how that occurs, and there's some new good work on that. But again, it's as well as like it doesn't have to be that way, right? In the sense, like, there are plenty of mammals with smooth brains,
yeah,
right? And so yeah, it's speaking to something that needs to be understood. But yeah, really really interesting. Sorry.
yeah. So then we have this human assembloids reveal the consequences of CNA1G gene variants in the thalamocortical pathway. So this is talking about the thalamocortical pathway and some of these human assembloid models of it.
Yeah, and this was a hot kind of this was a hot gene to look at, and I think we did a lot of we sorry, my PI had done genetic analysis of a lot of the imaging subjects that he had. So we did a lot of these kind of single allele or single gene studies. And CAC was one of them. I forget what, well, they'll probably say more about it here, but yeah, this was kind of looked at a lot by psychiatric neuroimaging people.
Okay.
Yeah.
Yeah. Okay. Then Paola posted something on sort of these. It's an article on these creatures occupy third state beyond life and death, and then Morgan this is on the Xenobots. So this is a article on the Xenobots. Yeah.
Right.
But like the way the headline is, it's kind of
that's I think that was also my surprise, because I was just like, what are they talking about?
Yeah.
And it's just like, oh, yeah, it's not exactly a third state, right?
Right.
It is what's the way the Levin puts it, or Bongard puts it? It's just like when not given their tissue, when not given their natural tissue context or something like that.
Okay.
Like but it emphasizes the agential material, and this is that they have the potential to form new collective structures.
Yeah.
Right.
So you're basically taking it out of its tissue context,
as opposed to this clickbait title.
Yeah, and then putting it into a new context where it can maybe free itself to form new structures.
Yeah. Who wrote that? Who's the Science Alert from?
I don't know. Let's take a look. You got me to click, so it was successful. Peter A. Noble.
They succeeded.
So yeah, it's basically about the Xenobots, and this, so this is, and just talking about biobots in general. But yeah, it's like, so these these are just basically organoids, and having this sort of reflection on what they are. They're not sort of the typical developmental system, because we don't have the tissue context, but we can take it out of that context and do interesting things with it. So
in their defense, the person writing this article typically looks at, we are researchers who investigate what happens within organisms after they die, which is kind of interesting.
Yeah.
I mean, for proper science reasons, not just but yeah, still nothing to do with xenobots.
Yeah. This is a workshop on data-driven discovery at the Allen Institute.
Right.
This
Yeah.
is
Yeah. They were just pushing one of their meetings.
yeah, AI and modeling in biology. So
Yeah.
this is I think we talked about this paper before. This maybe in maybe in VivoWorm, and that is how to build an artificial cell, or how to build a
This is
cell with artificial
Chan
intelligence.
Zuckerberg. Yeah. I mean, again, they were kind of pushing their own agenda here.
Yeah, how to build the virtual cell with artificial intelligence, priorities and opportunities. And we talked about this in a DVOR meeting where I kind of went through some of the vision that they had. And it's very similar to what we've been talking about with foundation models and using the standard, approaches of the day in machine learning to approach, trying to model a cell. So, I mean, this is not...
Yeah, and and sort of looping back to the Human Genome Project kind of discussion. We need like I guess one of the reasons that I post these is that Allen Institute and Chan Zuckerberg are they needed to reach a certain size or have a certain funding to be able to do these kinds of things, right?
Yeah.
It's which sort of speaks about it's kind of interesting again thinking back to the Human Genome Project and kind of like what it took for Ventnor to go alone, which is to say he didn't do it without a lot of resources.
Right.
Right. And and yet at the same time, this is sort of beyond any single PI, certainly any kind of ECR, right?
Right.
That this is very difficult to put together, and it speaks to kind of like the difficulty in getting kind of like the software engineers you would need, to getting kind of the research staff you would need, and having a kind of really kind of like center long funding horizon, and and certainly funding stability to be able to get anything like that done,
right?
And as well, sorry, and just to follow up on your post of that Professor Dave video, which I thought was really good, but it was like probably is true in science in a lot of science, right? Which is that like because you need these big science projects, that of course there's going to be people complaining about this or that is missed, something like that, because there's a kind of there is anyway, there's some scarcity, and then there's arguments about how that scarcity is used.
Yeah, it's definitely an agenda setting. Like you say this is what we need to do, but of course, I don't know if AI models, because when we think about modeling the cell, we need to have metabolic models, network models, a diversity of models that really kind of fit what we're dealing with. And then there's this approach where you say, well, we can just plug it into AI and find interesting things, which isn't necessarily going to help you, because the reason we use those kind of models that we use in like not a foundation model context is because they sort of approximate what's going on biologically. So
yeah. And as one of the things that came out of that Foundation Models Michigan series was just that, like, again, how much of our current AI hype is driven by basically GPUs and all the text on the internet.
Yeah.
Right. like it's cool. It's everything, but like it doesn't necessarily generalize well.
Right. So yeah, let's move on. I want to kind of wrap this up I mean, we've been looking at a lot of the papers, but
Yeah.
so we have this paper, Multicellular Artificial Neural Network Type Architectures Demonstrate Computational Problem Solving, and we can come back to that later in the meeting, which I have it in the folder, so we might talk about that. And it's basically where they're getting these, I guess, these bacterial cells to do computations, and they're getting them to make these binary decisions, basically giving them a question. And what they do is they have, like, in the bacterial cells, they'll express something from a genetic circuit, and then they'll take that and use it as the computation. So you can do these in bacterial cells because the bacterial genome is relatively simple. You can use like a GAL4 UAS system to get your bacteria to express certain things, and you can then drive it with like a stimulus. So you can actually do computation much easier in, say, like a bacterial colony than you can in, say, like a eukaryotic organism. But what they do here is interesting, and maybe we'll come back to this later. Then we have this Sergiu Pasca on assembloids. This is just,
if you want to move down, and he has like 3 papers, but I think the bottom one is four assembloid.
okay. Yeah, this is kind of, I don't know why it's dark like this, but the title is Assembloid Model to Study Loop Circuits of the Human Nervous System. This is a bioRxiv paper. So yeah, it says, I don't know if there's a link to the.
Oh, sorry.
Okay.
Let me see if I can. Oh yeah, here. I'll give you a preprint of bioRxiv.
Okay, yeah, that's good. So this is one of the models he has here: the neural organoids, neural assembloids, and a disease model. So the neural organoids are basically these different sort of organoids of a tissue type. So you have muscle organoids, glioblastoma organoids, striatal organoids, thalamic organoids, basically different parts of the brain or tissues in the brain that are, you can build an organoid, which is just this ball. And then the assembloid is where you put those organoids together and you assemble them into some structure. So the forebrain assembloid is subpaleo organoid and cortical organoid. And you're putting together 2 tissues, getting them to functionally integrate. And then you have migration assembloids and projection assembloids, and the neural assembloid. Basically, you have things that can, where cells can migrate between the 2 tissue organoid types, or they can project their projections into each other. So that's what we have in the corticostriatal assembloids, the corticospinal muscle assembloids. So we're building pathways now from these tissues. And then you have these disease models, so you can take the assembloids and look at deficits in migration, deficits in connectivity, which come from the inability to project into the proper targets. So you can study these different, pathways, but also disease models where those pathways are disrupted. That's really cool. That's a nice way of kind of thinking about that. And then, then Morgan posted this paper. Yeah.
And that's just that's the actual study. Yeah, or that's the.
Okay, so this study, is sort of what they're doing in that figure. So yeah, and they're building these recursive loop circuits.
Yeah, and this, I mean, I believe that's the most, or the largest number. I'm trying to think. Madeline Lancaster's advisor in Vienna. Noblish. Okay. Sorry. I'm getting old. Noblish has a 3 organoid assembloid.
Okay.
I think that's dopamine focused. So it has like a basal ganglia component, cortical. I forget now, but anyway, so this is cool in terms of what they're doing, and again, Sergiu's the head of the brainorganogenesis.org group that does the really cool workshop that I'd really like to participate in terms of covering their protocols. Yeah.
All right. Yeah, that's good. So that's our channel, our Brain Organoid channel. We've caught up on it. So thank you, Morgan, for doing a lot of the posting there and getting us those resources.
Absolutely.
And I hope that we can follow up on a lot of this. All right. So we have some posts in the chat here. Yeah. So there's just kind of, yeah. There's any follow up on, yeah.
Yeah.
So I don't know if Jesse wanted to give any update, or I don't think he had a chance to.
Hey, yeah, I was also when you were talking about like how much success is based off of like GPUs and what's up with available internet data and stuff like that. I was trying to find a good version of the like Hack the Planet gift that we love sometimes, just in the sense of like I think that's a really great question in many ways, and I did appreciate the discussion earlier on the Human Genome Project, and Hussain was here, and sort of the different points that everybody brought up about that. As far as updates go, a lot of work behind the scenes this week. Congratulations to Jen. She's made it to the first round of a certain fellowship that might lead to projects for like mental health related stuff. It's a little bit more, well, it depends. Depending on how things go, it could lead to a little more research project than other things. But it's sort of to be determined on what will happen there. But it's good to see. One of the things I'm really curious about right now and finding myself in like going into is sort of these grants, fellowships, opportunities, and I'm not really able. I know I mentioned in like one of the admin channels about like, hey, let's start going over these, what's coming up, what can we target, and what's available. I'm not really able to. In the past, I would lead through like a big chart about that, and I can't do that right now. But I am finding myself increasingly in that space, and I really think most of them are much smaller things, or more like individual based, and here's how you can find this and that. But to that end, excuse me, I never posted this in Slack. It was always saved as a draft. I'm actually also looking into automating a little bit of web scraping, some stuff around these topics, because I know we have such niche things that it's sort of an interesting game to try to find what's happening and where and who can apply to it. And then specifically for, I would say in sort of a Joe Pro lens, and it fits this lab too, but even more for Joe Pro, like things that are like very intentionally open to I'll use the P word of progressive, and I don't necessarily mean everything that it means about that, but like progressive in a people open to doing science in new ways or in open source ways or in different ways, and finding what's available in those spaces for opportunities is interesting. Like there's actually quite a bit. It's just, especially for things that are like, there's a bunch of new organizations popping up all the time that are trying to fund stuff differently. It's just they're new and it's sort of on the cusp anyways. So for anyone interested in that, please reach out to me in Slack, or if you're seeing this wherever wherever you know me from, because I'm very interested in. I feel it's like old school not quite gangster, but sort of old school like fixer role of like getting people attached to opportunities and then helping them develop projects. Like that's I enjoy it, but it also seems to be this intermediary role that I'm finding myself in a lot of ways lately. So for people who have advice about that or who are looking for stuff or who know where to find stuff, let me know. It's been interesting being here in Boston in that sense. And I'm not really spent a lot of time in the Bay Area out west because I know there's different sort of geographic things going on there and nature's for all that stuff. But it's just been an interesting I don't know, couple months that's ramped up into completely by chance finding an opportunity that I think Jen has a really great shot at, which would help her and further like in turn do some things for the lab, but also like it's sort of like getting your first research funded sort of research project opportunity that could further a career in doing more research or work or projects or initiatives kind of things, and that's cool. So congrats to Jen for that. I've applied to a few things myself. One of them is kind of simple. One of them is more involved. We'll see. Also, I applied to the Foresight Institute Fellowship a while ago, and I'm assuming I didn't get it because there's been no community. I haven't seen anything about it. Maybe I'm missing it, or maybe I'm just not cool enough or forward thinking enough. But like I don't know what that is. I had an interview about that like a month ago, and I think I struck out. But that's fine if I did. I think I know like scientists or people in maybe graduate programs kind of get PhD students get this eventual push like, oh, here's how you pursue funding opportunities. But for people that aren't in that or who aren't doing that but still want to do research, it's definitely its own like, approach and skill set and all that stuff. So that's a very broad, higher level research coordinator-ish update. Outside of that, there's a number of things in terms of like basically applied data science that I'm looking into for various projects in the lab. Also potentially combining with, I guess, sort of meta science approaches, which meta science is sort of my current word for, sort of quantified-ish takes on philosophy, history of science, the Meta Science Alliance, and I've been trying to get a meeting with ICOR, the I think something for open source research or open research group. I did a few things, particularly some weeks ago with them. I attended an event. Is what I should say. So I'm curious to do more in that space as well. And yeah, we also had we finished the Robbins on Bergson memory paper this week, which was nice in our other meetings. I appreciate that discussion too. That's kind of my updates, I suppose.
Yeah, thanks for the update. So yeah, you talked about your applied data science and meta science interests, so that's the thing I want to get to next. So you posted an interesting thing in the Slack this week, and I wanted to go over it. So this is the UCSD map of science, and this was I guess a paper by Katie Borner, who does a lot of visualization and et al. And they did this, I guess,
like 12 years ago, but yeah.
So yeah, it's 2012. And so this is the global maps of science that can be used as a reference system to chart career trajectories, the location of emerging research frontiers, or the expertise profiles of institutes or nations. And then, kind of showing this UCSD map of science and classification system. So this was based on using 7.2 million papers and the references from Scopus, from the Web of Science, Arts and Humanities citation indices, and more. And so this is where they, built this corpus and they kept adding to it. And so, to our knowledge, it's the first time that a widely used map of science was updated. A comparison of the original 5-year and new 10-year maps and classification systems show, one, an increase in the total number of journals that can be mapped by 9,409 journals. So there are more journals. A simplification of the map by assigning all but 5 highly interdisciplinary journals to exactly one discipline, and then a more even distribution of journals over 554 subdisciplines and 13 disciplines when calculating the coefficient of variation, and then a better reflection of journal clusters when compared with paper-level citation data. So this is pretty hardcore science visualization of like basically different fields and how those things kind of fall out. So papers are being published in different fields. Sometimes they're interdisciplinary, but they fit into a certain set of fields. And then you can see that, different fields are connected to one another through, co-citations and publications. So this is the map here. This is a visualization of the UCSD map. On the left, there's this two-dimensional Mercator projection, and then with 3D spherical inserts at the top, and then this 1D circular map on the right. So basically, the way they do this is it's a network, and they have to project it in some way. So, it's on this sphere or on this circle, and they've taken this network and they've flattened it out into this two-dimensional view here, this network right here, where you start with kind of, it's sort of toroidal. So, the left side connects to the right side and the right side connects to the left side. So that's how you're supposed to interpret that. But it's like a map of the world. You have math and physics on this side, CS and electrical engineering down here, which is pretty close to the math and physics cluster. You have chemical engineering, mechanical engineering, civil engineering here. You have earth sciences connecting down here and chemistry connecting up here. Then moving out further, you have biology, biotechnology, infectious disease, and medicine. Those are all expected to be kind of in a group together, connected to earth science in the case of biology, and chemistry in the case of biotechnology. Then you have brain research, which connects to health professions and medical specialties, but also the social sciences. And then the social sciences connect to math and physics on this side, and then the humanities are out here kind of connected to social science. So it's not surprising. I mean, we kind of know if we just read the literature and how things are referenced. I mean, from our discussion today, we had a very, sort of math and physics / biology / chemistry heavy discussion with some brain research and medicine. These are all kind of connected in different ways. But also, when we have our discussions about like AI ethics, it's kind of more social science oriented, maybe some brain research, but more humanities. So that, and we know kind of intuitively that's the case, but this kind of shows the network topology of it. So, what are the insights we can learn from this? Well, I mean, we can learn kind of how different publications are connected. We know what fields look like, but, the publications are really the thing that define the fields more than kind of university departments. So university departments just house people under a name, but a lot of times people do different types of research, and it connects to different things. And so this is a way to identify, not only sort of the boundaries of a field or what people are doing in a field, how they're connecting with other areas, but also kind of the opportunities for interdisciplinary collaboration. And that's not to say you couldn't have an earth sciences humanities collaboration. It's just not shown in this map, just in the way it's built. So that's okay. So this is interesting. And then on the right side, we have the circular map that kind of shows the domains as they're sort of aligned with one another. So there are adjacencies between different fields. And, that's what that's showing. And then this is their SciVal, which is their interface So you can actually look at specific papers or specific groups of papers in these, I guess, competencies. Each circle represents a competency of the University of California at San Diego. So they're using a specific university, and they're looking at these competencies at the university, and they're looking at the papers that kind of fit into that competency. And then their subject areas. So each of these competencies have contributional, the subject areas contribute to this through papers, and you can see that breakdown on these circles with these lines. There are different colors which correspond to these subject areas. This is a really interesting interface. And then this is a graph here where they actually have a fractional journal count for each type of journal. So they kind of show this, and then they show this. This is Indiana University's Vivo, which is like a research platform, and you can see faculty sign up for Vivo, and certain universities use the system, and it just shows you kind of the different activity in different areas by the number of publications. So it's really interesting. I don't know if this is good for identifying different fields and how papers fit into the fields. Unfortunately, it could be used as sort of like an evaluation metric, which has a lot of negative implications, some positive, but mostly negative, as far as I can tell. But I mean, this is nice work. So yeah, thank you for posting that, Jesse.
Yeah, it was quite interesting. Also interesting that it's my university that did that. I'm curious to look into it a bit more, and as I said, there's sort of this interesting space between meta science, complexity, data science, and this kind of stuff that I'm exploring. And I really wonder if there's other. I'm curious what modern work in that space is doing, or what if the researchers have done other things. I really haven't followed up on it, but we'll see. We will look into that.
Yeah, it'd be interesting. I mean, I don't know if I'm sure they've updated their database and their methodology a little bit, but so I think I'll talk about this this set of work. This is a collection of papers mapping reward functions to the brain. So this is a post on Blue Sky by Blake Richards, who's an AI person, neuro AI person, in fact. And so they say, I think one of the most important challenges in neuro AI in the coming years will be mapping to different neuromodulatory systems, mapping the different neuromodulatory systems onto different internal reward functions. And so papers like this are kind of the groundwork we need for that. And then there's this link to the paper, which I will then show you next. This paper is Dopamine and Norepinephrine Differentially Mediate the Exploration-Exploitation Trade-off. So this is, we talked a couple weeks ago about the research agenda for neuro AI, the different interesting questions in the field, and kind of what the good kind of sub-disciplines are, or sub specialties are. So this is maybe kind of one take on this. So it's basically, if you're looking at neuromodulatory systems, can you map those to different internal reward functions? And if you can do that, then you can simulate maybe some neuromodulatory system, build a simulation where you can then replicate these reward functions and then fit that into a model of, like a neural network model, and then have a better sense of like how reward, sort of informs behavior. So that's kind of where this is going. And we talked about dopamine a while back, and how, there's this sort of idea that dopamine is a reward molecule. And, it's true that it's a reward molecule. A lot of the neuromodulatory functions of dopamine are less clear. So, it's not like a straightforward, you get more dopamine, you get more reward, and that's kind of like, that explains everything. It's a little bit more subtle than that. So this is a nice kind of insight. And this is the paper. This is, I guess, Blake Richard's paper, so that's why he was advertising it. But and so this paper is called Brain-like Learning with Exponentiated Gradients. So okay, so let's go into the abstract. The abstract reads, computational neuroscience relies on gradient descent for training in artificial neural network models of the brain. The advantage of gradient descent is that it's effective at learning difficult tasks. However, it produces ANNs that are a poor phenomenological fit to biology, making them less relevant as models of the brain. So basically what happens is that gradient descent is used as sort of a heuristic for learning difficult tasks. If you climb this gradient or descend the gradient, it's basically a way to discover like an optimal solution. And then that's the idea of your neural network, is it's trying to climb these gradients or descend these gradients and find the right, find the optimal solution, the global minima or maximum for that matter. So that's the logic behind things like reinforcement learning or also genetic algorithms. And you use these biological metaphors to sort of, climb that gradient or descend that gradient in a way that's differentiable. But of course, biology doesn't necessarily do that. We don't have like gradients in the brain. We don't store gradients in our brains as representations. We store maybe, we can optimize our neuronal networks. We can have, if we're dealing with an organoid, we can optimize those neuronal networks. There's a lot of signaling that goes on that might go in a specific direction, but we can't necessarily, there's no gradient. Maybe there's an energy gradient that is, something that the brain is constrained by. Like, it can't violate the laws of physics. But to say that there's a gradient that network is conscientiously consulting as it learns is not the case. So it's a poor phenomenological fit. So specifically, it violates Dale's law by allowing synapses to change from excitatory to inhibitory, and leads to synaptic weights that are non-log normally distributed, contradicting experimental data. So in using gradients, we often, kind of have to bend the rules of biology and, kind of get the answer that because in an approximation of learning, it's fine. But if you're trying to map this to the brain, it's not fine. Here, starting from first principles of optimization theory, we present an alternative learning algorithm, exponentiated gradient, EG, that respects Dale's law and produces log normal weights without losing the power of learning with gradients. So we can get these gradient-like things and get our system to conform to gradient-like things without, kind of putting this artificial model onto the system. We also show that in biologically relevant settings, EG outperforms GD. So this is where, I guess biologically relevant is, where you have actual biological networks that are learning things, including learning from sparsely relevant signals and dealing with synaptic pruning. So this is where you're getting changes in the connectivity, or you're getting sparse signals to learn from, and you have to kind of generalize things and make judgments about things given very little prior information. Altogether, our results show that EG is a superior learning algorithm for modeling the brain with ANNs. So this is where, we want to model the brain, not necessarily model data. And so we have to kind of think a little bit differently about this. So, people have been, aside from like machine learning, people have been applying gradient descent to computational neuroscience studies, and here's some citations here. Gradient descent is leveraged in these studies because it is very effective for training artificial neural networks, even though it is not considered biologically plausible. Moreover, it is clear that gradient descent is phenomenologically different from the learning algorithms of the brain. So first, gradient descent prescribes synaptic changes that violate Thales' law, or changes from synaptic inhibition to excitation. So it's just changing the sort of the sign of that, which is important if you're talking about learning and knowing how that's regulated, right? Second, GD does not produce synaptic weights that are log normal. And so this is just the distribution of synaptic weights, and this is something that has an effect on how they perform, especially with response to real-time behaviors. And this has been observed. Log normal distribution of synaptic weights has been observed across species and brain regions. That means that the activity is not like distributed across the network widely. It's distributed in like maybe certain places in the network. Even if we set aside concerns about biological plausibility of gradient descent, these phenomenological differences motivate the search for an alternative, more brain-like learning algorithm for training models of neural circuits. However, from an alternative algorithm to be better than GD for computational neuroscience, it should be no less powerful at learning than GD, ideally better at learning in biologically relevant situations. So this is, basically the reason we use gradient descent in the first place to, say, learn difficult tasks is because it is fundamentally normative. So it provides the greatest improvement in a measure of behavioral performance with the smallest changes to the synapses as possible. So we don't want to change the synapses a lot. We just want to change them in a way that's, we can understand, but also in a way that doesn't disrupt other behaviors, which is, not necessarily the way biology does this. Biology can change synaptic weights quite a bit, but then you end up with behavioral performance that is, maybe adaptive, or maybe you can have different behaviors emerge or things like that. So what does it mean? But what does it mean to measure synaptic weight change? Here we reanalyze this core theoretical assumption and show that gradient descent does not align with biology. So in measuring synaptic weight change and understanding how this is occurring, they find that gradient descent does not align with biology, partially because it is based on a measure of synaptic change that is agnostic to changes between inhibition and excitation. Using this insight, we explored a related but different learning algorithm, exponentiated gradient. And this is from this publication, Kivinen and Warmuth in 1997. Like GD, exponentiated gradient is also derived from normative first principles and leverages gradient signals for learning, but is based on a measure of synaptic change that adheres to Thales' law. This works as well as gradient descent for deep learning, which rules out the common concern that brain-like algorithms do not scale to hard tasks. Okay, so this is, let's see if we can find some figures here. This is just showing, these differences between the two types of, gradient descent and exponentiated gradients, and how we get the synaptic distance penalty between time points. We also have some of these task responses and the training response. And so we can base exponentiated gradients solve the mod-cog suite of tasks, as well as gradient descent, but with more biological weight solutions. That's figure 1. Figure 2 is exponentiated gradient finds weights that are easier to prune and easier to retrain after pruning gradient descent. So in learning, you often prune synapses and you grow new synapses, and this is to reconfigure the network, not through necessarily weights, but like just pruning the connections and establishing other connections. And so exponentiated gradients allow you to do this more easily, and this also has implications for developmental AI as well, because in development we get a lot of weight pruning or synaptic pruning And that's a key feature of sort of the development of things like the visual system. Figure 3, exponentiated gradients outperform gradient descent when relevant inputs are sparse. So this is where we have these sparse inputs, and we get this better performance. And then in figure 4, continuous control with noise is better than exponentiated gradient. So this is where you get this, I guess this is continuous control with noise. And you're using exponentiated gradient, and it's better, I guess, than using gradient descent. So continuous control with noise is where you basically have these naturalistic behaviors, like moving towards a target, or getting feedback from the arm versus and filtering out the noise feedback that you might get, or the noisy feedback. And so this is I guess a robot arm, where you're measuring its starting point and then moving towards a target. This is the recurrent neural network that's sending muscle commands out. So this arm is kind of moving towards a target, and it's sending the muscle command, so it knows how much time, how much control it needs to, how much modulatory control it needs to reach the target successfully. And then, of course, you have to train your recurrent neural network. So before training, it's pretty sloppy. After training, it's pretty precise. You get arm feedback and noise feedback, so you get the feedback from the arm, and it has to be filtered out from noise, which is where you get like inertia and things like that. And so it's able to sort of converge on, some sort of target solution. And then this is where you look at the paths that this controller takes to the different targets from the origin. And you can see that, and this is a classic kind of plot that they use in motor learning, where they kind of look at the path that the end effector is taken to the target. And you can see that in this is exponentiated gradients, this is gradient descent. So gradient descent, there's a lot more variability, a lot more noise included in the movement. So this is almost like, someone with a movement disorder trying to move to a target. And then this is exponentiated gradient, which is a lot smoother. And there's only some variation at the target location, where it's trying to find the exact target. And so the paths, however, are smoother. And so you can see this for 3,000 and 6,000 updates. All right. So I think that's, some nice work on, there you go, Morgan.
Sorry. I did. I had to step away for a second. No, really interesting. Yeah. I mean, a great example of like you said, or like he said in his paper, not trying to do data analysis, trying to create a brain,
right?
Or like an artificial brain. So yeah, really, really interesting in how kind of like a simple property created, or a simple, a somewhat simple difference gives you all these, brain reproducing features or behaviors. Like great example of neuro AI.
Yeah.
Or, whatever we want to call it, brain AI.
Yeah.
Yeah. So very cool, and not surprising. I'm pretty sure. I wouldn't be surprised if he was one of the Montreal speakers.
Yeah, I think he's in Montreal actually. Yeah.
At the main meeting. Yeah. And just wanted to add. I was just gonna say, what were the meetings this week that were of note? I'm blanking. I had one like 20 minutes ago.
Okay.
And just again, very happy. My daughter was complaining about my YouTube use, and I was pointing out to her that I'm not exactly on TikTok. I like the fact that YouTube gives me access to the world's scholars
Yeah.
and the talks that they are giving. And anyway, some great meetings that were all, that they posted all their videos online, and I will check, but I usually drop them in the Slack.
Yeah, we can revisit those as we need.
Yeah. There was something. Oh, I mean, Santa Fe Institute dropped a bunch of videos, but I don't know if they were super recent. But I dropped one in Frontier Mac just because it seemed like it was kind of patent related. It reminded me of the old paper from Mark Badeau on patents, and was it like Norman Packard or something like that? Yeah. Anyway.
Yeah,
but
take a look at that.
Oh well, it's and a great talk. I forget what he called it. It was a really interesting Santa Fe Institute talk about, like, are we on the way to Cincinnati?
Okay.
Which is a funny talk title. Anyway, social psychologists, experimental psychologists, but also a comedian, a stand-up comedian, and so they when they were introducing him, they said something like, he's this professor and that, got his degree here. He is also on IMDb with a movie that has, that is achieved 3 on the Rotten Tomatoes scale. Like, anyway, it was really good talking about a good critique of experimental psychology's progress. Anyway.
Okay, yeah. We'll have to take a look at that. Okay, so this is the paper.
The competition.
Yeah.
Right.
So this is multicellular artificial neural network type architectures demonstrate computational problem solving. So we talked about this earlier, where we have this. This is the visual abstract. And I'll zoom in a little bit here. So this is where we have these bacterial cultures, where we have bacteria that are doing, I guess, quorum sensing, and they're expressing genes that, they can express genes in a way that's almost switch-like, and you get these, you can also express phenotypic states in this way, and then they can do computations, so they're actually, each flask can do certain gives certain sets of answers to different problems that you pose to it. And then, the solution is the answer that it gives. So it's, it seems a little magical. So let's get into the details of this. So the abstract reads, here we report a modular multicellular system created by mixing and matching discrete engineered bacterial cells. So in each of these flasks, you have certain engineered genetic circuits that you can then trigger, and you can get them to express different things. So, if I create a strain that has one construct in it, I can get it to give a certain answer, and if I have another construct, I can give it another answer, which is, not necessarily like surprising, because I've engineered the cell that way, or the gene circuit that way. But, we can then combine them together to solve multiple computational decision problems. So you have these systems where you grow these bacterial cells and cultures, you engineer them for certain competencies, and then you mix and match into different flasks, and you can get them to solve more complex problems. So you see that it's giving each flask a problem. So this problem is for a prime number. This other problem is 7 factorial divisible by 7 times 7 plus 1 divided by 2. And it's either no or yes. And then, of course, you could combine these into a larger problem and give solutions for those components of the problem. The modular system is based on a set of engineered bacteria that are modeled as an artificial neurosynapse. So basically the analogy they're using is that I can use a gene circuit in each one of these bacterial cultures as an artificial neural synapse. So if you take different strains and you put them in a co-culture, meaning I'm taking like some cells out of the first flask, some cells out of the second flask, and I put them in a third flask where they're in mixed population. If I do this, I can form a single layer artificial neural network type architecture that can perform computational tasks. So essentially you're getting answers from these cells and you're forming this sort of artificial neural network type architecture. Basically, each population of cells is a different layer in that neural network. So it's like kind of going way back to the early days of neural networks, where people would analyze the layers and, they're hidden layers, of course, but we kind of know what the hidden layers are here. As a demonstration, we constructed devices that function as a full subtractor and a full adder. So you have devices that can, subtract and devices that can add. So a device would be like these co-cultures that are basically these layers. And then you can kind of model the layers as a very simple neural network, or model the answers that are given as a simple neural network. So you have these devices that function as a full subtractor and full adder. The system is also capable of solving problems such as determining if a number between 0 and 9 is a prime number, and if a letter between A and L is a vowel. So it's trying to identify different stimuli in a certain way. So I'm not really sure how it's asking the questions. I guess that's my problem. I can't get access to the paper, but basically it's solving these problems, or it's posing these problems, and the gene circuit is solving them. And then you have this co-culture where it solves a bunch of these questions. You might parse the question. It's kind of like a large language model where you prompt it in different ways and you just find the right prompt to get it to learn in that way. Finally, we built a system that determines the maximum number of pieces of a pie that can be made for a given number of straight cuts. This work may have importance in biocomputer technology development and multicellular synthetic biology. So, I can't access this right now.
Yeah. And I mean, I take it you already checked Sci-Hub.
Oh, I didn't check it right now, but
I'm just kind of curious because this relates to another paper in terms of it's doing a Cloudflare check. No, it doesn't have it. Okay. So very in brain organoids, just past the next paper, so the tissue talks, this Ron Weiss tissue talks from October 16th, programmable organoids guiding cell fate with synthetic biology and neuromorphic circuits. Okay. Was what it was very similar. What Ron Weiss? Let me see if I can find his. If I can, I think he's got a good website.
Okay.
Just in terms of somebody definitely trying to develop that same kind of technology, if you will, or like here, Weiss Lab. Not necessarily super interested himself in terms of the. I'll just drop it at the bottom of Organoid channel. But somebody who's definitely working on those kinds of genetic circuits, and genetic programmable logic or synthetic bioprogrammable logic. Yeah. Just reminds me a lot of that.
Yeah, so this group is doing this and trying to do this in neurons or in bacteria, or what are they? Let's see their publications.
Or I don't know, Vice.
Yeah.
It's cellular.
Okay.
Like not neurons. Yeah. So depending on what. Yeah. Like he'll use whatever model or whatever organism makes the most sense, but potentially just E. coli or yeast or something like that. Like and unless it's medically related, and then it's like, oh, can I do this in say human cells or something like that?
Yeah. Yeah, it looks like he has a paper on synthetic genetic circuits to uncover the OCT4 trajectories of successful reprogramming of human fibroblasts, which is interesting because that's definitely mammalian cells.
Absolutely, like he's absolutely doing human research too.
Right.
They might proof of concept stuff and simpler stuff. And yeah, like it was just a really nice integration of kind of synthetic bio and organoids. Right?
Yeah.
Yeah. But you could absolutely see how to use his work to do biocomputation.
Right.
And I kind of like those groups where it's just like that's not necessarily their focus,
right?
But so sometimes it's like they come up with even better solutions.
Yeah.
And potentially more scalable ones. But this is a very different kind of biocomputation from again, I always think of, or I'm so old, I think of the kind of DNA computing, like are there ways to do kind of parallel computation with biomolecules that could be computationally, you could achieve, quote, supremacy over classical computing algorithms, right? And these are not, that's not this focus.
Right. All right, that's great. So yeah, well, thanks for attending. I think we had a good session today. Reviewed a lot of stuff, and hopefully we can move on the organoid stuff soon, and get some.
And do you think Stefan will be enough for the open source symposium, or the little part of the active inference?
What do you mean? Like, would he be involved, or? I don't think so. I mean,
no. Just as like if he's the only speaker.
Oh, you mean, yeah, that'll be fine if he's the only speaker. Okay. Like, yeah, you're sending out the invites. Yeah, I just want to make sure we get one person, but we could have two if we need to.
Yeah. I don't know if if for Ankur and Yarrick, that's just that's not a time frame on which they are willing to move.
Oh, okay. So we're kind of down to Stefan.
I certainly haven't heard back from him.
Okay, yeah. Well, okay.
Yeah.
All right. Thanks.
Okay.
Talk to you later.
Take care.
Take care. Bye.
Bye.